And we're very fortunate not only that we have Dr. Patrizia Casaccia at Mount Sinai, but that she's going to spend some time telling you about her research. Dr. Casaccia is a highly recognized and productive neuroscientist. She is a professor of neuroscience and genetics and genomics at Mount Sinai. She acts as the director of the Center of Excellence for Neural Repair in Demyelinating Disorders at the Friedman Brain Institute at Mount Sinai. She's worked in a number of areas in her laboratory, in particular looking at myelin formation during development, in ways of repairing the demyelinating lesions, and in particular, she's had an interest in how factors modify our underlying genetic makeup. Most recently, Dr. Casaccia has been honored with the receipt of the very prestigious Jacob Javits Award from the National Institutes of Health in recognition of her work. Dr. Casaccia is going to talk to us about research in MS. Can the course of the disease be modified?
So good morning, and thank you so much for being here. I just have this task, right, to try to familiarize you with the research and hopefully excite you. And I'm going to be showing you, hopefully, if they work, some movies also. And I hope that you have no problem also seeing mice, because oftentimes we have to work with mice in order to test some of our experiments. So with that said, I just wanted to briefly tell you a little bit also why I chose research. And I come from Italy, and in Italy there is a very high prevalence of MS. And as a medical student, I was working in an MS lab recording evoked potential to other students. And this is how I found out that one of my best friends had MS. And then my aunt had MS, and then my cousin had MS. And this is why I dedicated practically all my life to research on MS. So the major question that we have today is whether the presentation works. And so one of the major questions that we have, and perhaps many of you are familiar with this, is after you get a diagnosis of MS, the question is, now what? Why me? What can I do? How does it work? What can we do, right? And so what I think is going to be interesting for you is to try to understand a little bit what we do every day, right? I've been doing this now for 20 years. And what can you do in order to perhaps modify the disease course? Now, this is just a cartoon that shows you here an oligodendrocyte in yellow with the myelin that wraps around the axon. And the axon is a part of the nerve cells that's essential to move the information from the brain to a muscle or to a sensory cell. So this is the way we think, this is the way we talk, this is the way we walk, this is the way we feel. And this is how it looks if you take a slice of the brain where all the white part is myelin, the part that's kind of pinkish is gray matter. Now, if you see what happens in MS, you can see the myelin becomes all red and inflamed, becomes attacked, very badly attacked by immune cells. And what happens then is instead of having all these white areas, you start seeing these regions pointed out by the arrows where myelin is lost. This is a plaque. This is what in the MRI then we can detect as a specific blob. So the Multiple Sclerosis Society had this big campaign about what can we do. We want to stop the disease, we want to restore, and we want to end. How can we stop it? And many of the people, and many of you, I'm sure, are very familiar with the concept of stopping the immune attack using immunomodulatory drugs, using interferon, using Copaxone, using Tysabri. And what all those drugs are doing are stopping the immune cell from either entering the brain or attacking myelin. What we have less material for is how, assuming that myelin is damaged, because when you go to the doctor, probably most of your myelin may be damaged in certain regions, can we protect the axon? Can we make new myelin? And these are the questions that motivate us every day to go to the lab and try and try and try. So the idea is we don't know, not only we want to stop the immune attack, we want to restore function. We want to make sure that you can, feel better. And how can we do that? We can do that by blocking on one hand, trying to block axonal damage, and trying to find therapy to make new myelin. So once again, let's go back to the concept of axon. I showed you this little wire over there. Many times we discuss axons are like electrical wires wrapped by myelin. Clearly, if the myelin is stripped out, what happens is that the wire snaps. And when it snaps, the region of snapping forms this little ball over there. That's called a transaction. So imagine you don't have a wire anymore that's connected with another cell, but it snaps. So in order to protect this, we want to understand why does it happen? How does it happen? Can we do something to block this snapping? Taken from the spinal cord of a mouse, and what you can see is that first the axon is like a little wire, and then it snaps and it forms these large balls. When we try to go forward and try to understand what happens going from this wire to this snapping to then fragmented into these small parts, we notice that there are several little particles that are shown in there by the yellow arrow. Those are the mitochondria. So what are the mitochondria? And this is a little bit of biology 101. The mitochondria are these energy-making machines that we have inside all of our cells to produce energy, so that whatever we eat, all the fuel, starts making all these fluorescent particles that are called ATP and give the energy to the rest of the cell. Now, in the lab, we have machines that allow us to measure these mitochondria, that allow us to measure the energy that it is produced, like shown here. We can put the neurons inside this little chamber, and then we introduce it in the machine, and the ATP will provide light. And we can place these electrodes, shown here, as you can see, that are going to go very, very close to neurons, and then record the ATP that it is produced. So you would say, well, so what can you do with this kind of machine? And, we have the neurons into our brain. How can you measure that? Well, what we can do is we can use a trick. We can take neurons from rats, or we can take neurons from mice, and then expose them to the cerebral spinal fluid of patients. And some of you has very graciously agreed to participate to some of those studies by donating the cerebral spinal fluid from a lumbar puncture. So I wanted to show you, for those of you who might have participated, what kind of study we do with the cerebral spinal fluid after the lumbar puncture. What we do is that we take the serum, this is the hand of one of my collaborators, and we place on the neurons that are on the dish. And then using this machine, we can measure the energy. What we find out is that when we put the cerebral spinal fluid of a patient with MS for a short period of time, the neurons have to work much harder. They have to produce much more ATP. In other words, once you put the CSF of a patient with MS, all the brain requires more energy to live. So you are spending much more energy. Now, if we repeat this experiment on stressed neurons, when every day we stress them for like 15 minutes with something, we give a little bit of oxidative stress, then if we repeat the same experiment, the neurons, after they are exposed to the same amount of CSF from patient to the same liquor, they just cannot produce energy anymore, perhaps because they are exhausted. They have tried to produce, and they cannot. So what can we do, for instance, in this case? Well, we were discussing before about supplements, etc. In this case, everything that would help against oxidative stress, all these antioxidants, may be beneficial, for instance, in this sort of situation. The other important thing is that you have to think of our body. We are complex machines, right? We have the brain, we have the hands, we have the spinal cord that's very long. So how do you produce energy and we send it around? This little machine, these little mitochondria, have to move a lot because there are very long distances that they have to travel. And what we showed, and this is another group that showed the same thing, is that the mitochondria in patients do not move well. And I wanted to give you an example here. Now, in green, these little bright dots are these mitochondria that I was showing you in the healthy neurons. And what I colored here is the distance traveled in 5 minutes by one mitochondria inside the nerve. So now, just by yourself, compare all these traces that we label, by clicking every 30 seconds under the microscope. Look how much in 5 minutes the control mitochondria move compared to the ones that were exposed to the cerebrospinal fluid of patient. So we spent basically the past 3 years trying to understand this. Why? Why is it that if we put the CSF of patient, they do not move? If you put the CSF of normal people, they move. And we find out, and this is and we found out that this kind of Disney cartoon, if this long wire is along the axon, the mitochondria is this large ball, is carried around by this little molecule here in purple, which is a motor molecule. And what we found out is that in patients with MS, the motor cannot carry the mitochondria. So you say, well, this is very interesting. The motor cannot anymore carry the mitochondria, so what can you do about it? Well, we try to understand why cannot it move. What is that it's blocking this molecule? And we proposed a new mechanism. And we work with with a new biotech company in Boston to develop new treatments. So here I need some help with starting the video. So these are mice that are paralyzed in the lower limb and the tail on the left side. And on the right side are mice that were treated with some of these new drugs that we have developed that we started when the mice were paralyzed in the lower limb. So what this drug is doing is simply helping the mitochondria move, thereby restoring the energy to the hind limb of the mice so that even those paralyzed mice had the ability to move. Now, I want to also mention to you that clearly this is research, right? And many people have studied, well, how long does it take to go from when you discover something in the bench to something that it is available for patients? And some people have studied this timing, right? And some people say, well, it takes 20 years, 10 years, 15 years, 20 years. So we are trying to work more and more because, people are not animals, and we have to make sure that we guarantee that there is no toxicity of any sort. But the bottom line message is we have hopes. We have hopes that many of these treatments will work. We have hopes that many of these treatments can be translated to you, in few years, hopefully. But besides what we do in the lab, and besides every day, defining new drugs, is there something that you can do on your end? And one of the questions that I always had is, you hear a lot about, do you have the genes for MS? Or, we were discussing about the themes, and what does it mean to have the susceptibility genes? And there have been two schools of thoughts. There have been scientists that have devoted all their life to try to understand what kind of genes are in a MS patient. Other scientists that have tried to understand, does the environment play a role in this? How come that people in North America have more MS than people, for instance, in South America? Or perhaps it's both. So this map shows you some studies that were done by the Genetic Consortium, which is a lot of scientists from Canada, North America, Europe, and Australia. And what they find is that in these regions that have all this little red number, there are many people that have MS. So they've taken all the genes and they try to understand, is there a difference between the people that have MS with the people that do not have MS? And what they find is that many of these genes are involved with the immune system. So to have the susceptibility genes, it means you have an immune cell that's ready to attack. It's more ready than a cell than a person that doesn't have these genes to attack. It doesn't mean it's going to attack anyway. It means that there is something else that has to happen in order for these cells to attack. But there is another interesting thing that I want to mention, and there are many genes, 13 actually out of 57, that are linked to gut-related disorders. And there is a lot of discussion right now, very active, and I am indeed actually very interested in doing some serious research about diet, because I'm a true believer that diet will have a major role in it. And it's interesting that out of these 57 genes, 13 are related to gluten intolerance, which points to the fact that we don't know much yet, but we need to be careful and really try perhaps to understand what genes do you carry. Maybe if you carry those genes, you could be a responder to a gluten-free diet. Maybe you don't carry these genes, and maybe you are not a responder. But we can start developing new ideas, and we can start developing new trials to really test this hypothesis and really make sure we have solid evidence. For sure, the idea that the gut is related to the immune system and the brain is becoming a very hot topic in science. And just this year, there were several reports that the type of bacteria that we have inside our intestine do affect the immune system and do affect the ability for myelin. So why is it that even if you have those genes, you may or may not develop the disease? Well, there have been many studies, and in some cases, Michelle had discussed before, for instance, a radiologically isolated syndrome, and many additional cases where people, although have the gene, they may not develop MS. So this is the idea. The idea is that you may have two hits. The genetics defines your risk, but then a lot of other actions can affect this. For instance, diet, vitamin D level, lifestyle, especially smoking, that will interact with the genetic background, sun exposure, viral infection, especially ABV virus, and there has been some data about pesticides. So to divide, there are strong evidence, negative effects that have been backed up by solid science regarding smoking. There is no question. Smoking is bad for you. It's bad for everybody, but if you have MS, it's even worse. So absolutely, it is something that can worsen it. Infections are bad for you, low sun exposure, and vitamin D level, as you heard. And there is weaker evidence, although I think it is worthwhile for us to think very carefully to study it regarding the effect of diet. And in this respect, I wanted to share with you some example regarding the interesting distribution of high-fat diet and MS. It seems to be quite interesting that if you look at the distribution of fat consumption and MS, they seem pretty much to be distributed. So we wanted to test this in mice. And what we did is that we took two cohorts of mice, sisters. So they had exactly the same genes. And we put some sisters, like here, on a regular diet. Other sister in a high-fat diet. And on both of them, we gave them an immune booster against myelin. So after 11 days, as you can see, the mice that were, let's say, on a vegetarian diet in this case, they were still running around quite happy. However, the mice that were on a high-fat diet, this is the sister, they had clear paralysis of the tail, and they had clear paralysis of the back limbs. The only difference between these two cohorts of mice was the presence of high fat in their diet. So for sure we have a lot to do, but for sure there is also quite clear evidence from both epidemiological data and our mouse data that high fat is not good for you. We also did additional study regarding the effect of social isolation and chronic unpredictable stress. And although I don't have the time to show you all the data, here in red is myelin, and what you can see is that stress really decreases myelin. You can see that the stressed mice have much less myelin than the control, and here you can see that the social isolation has the same effect. So in this question, it is your gene, it's the environment, or both? I would say it's both. And why is it? Well, the reason is because our DNA is very, very long. It's almost 2 meters, and it has to be packed into a very small part that it is the cell. This is equivalent, if you think about this geography, this is the longest highway in the United States. Is this the longest bridge in the United States? It is as if you had to fit this 24 miles long bridge into the diameter of a ball. This is how much we have to compact and wrap around our DNA to fit it into the nucleus of the cell. So what happens is that the environment can act on this wrapping around by closing or opening the DNA, allowing some of those genes to be more expressed or less expressed. And this is how, with your own choices, you can modify not the genes that you have, but the way your DNA is wrapped around. And this is just to show you an example of the, can I have this movie going, please? So here what you can see is an example, right? So if we go inside the brain of the cells and some of the studies that we do, you can see that there are blood vessels, a lot of neurons around, and this is an oligodendrocyte progenitor cells. You go now inside the cell, inside the nucleus of the cells, there are a lot of particle movement, and this is the DNA that it is wrapped around the molecules. Now, depending on your lifestyle, you can affect the activity of all those machineries that are the enzymes that can put different groups. In this case, they are putting acetylated groups. They can put groups that activate genes so that some genes can be expressed. And you can see here there are many of them that are doing the jobs. You can also affect, and for instance, smoking would affect that. Diet would affect this. There are other genes, other enzymes in contrast, that can put different type of groups. And by putting these different type of groups, they invoke this tightening of the chromatin and wrapping even further so that at this stage, the DNA cannot be exposed anymore and the oligodendrocyte can survive and you can have a healthier, let's say, response to the immune attack. So this is how the environment works. The environmental evidence from vitamin D, from smoking, from diet, from toxin, they would work by moving around the DNA inside the cell so that certain susceptibility gene can be more expressed or less expressed. And so to end the talk, I just wanted to give you an example. So let's say that there is Mrs. X that is a female, so has all the typical characteristics of MS. Her mom perhaps had MS and had low vitamin D during pregnancy, was born in the month of May or early June, which means the mother had even lower level of vitamin D during the pregnancy because she was pregnant during the winter, lives in the northern countries, carries the susceptibility genes. Has this person for sure going to be develop MS or not? For sure, this person will have a hyperactive immune system. May or may not also carry the susceptibility genes for some of these celiac diseases. And now you have two choices. This person can just decide not to do anything, and therefore not check the vitamin D level, smoke, perhaps be a little bit obese, and choose a diet rich in fat, or can choose to measure the vitamin D, make sure that there is vitamin supplementation, make sure that there is no smoking, exercise, a healthy life with stress reduction, a lot of social interaction, and this for sure is going perhaps to have a much better outcome and of life in terms of the other possibility and the other choice. And finally, I wanted to conclude by saying, so these were the three major missions of the MS Society: stop, restore, and end, and we have been really trying to work on each of these components. We know that several pharmaceutical companies have been working on immunomodulatory drugs to stop the immune attack. Therefore, we are not working on this aspect, but we are working on developing new drugs that would support mitochondrial function. We are also working in other experiments that I didn't have the time to show you today on finding new myelin, new compounds that will help produce new myelin from inside the brain. But you can also do something from your end, which is, if you're smoking, for sure, stop smoking, check the vitamin D levels, exercise, reduce stress, and hopefully also have a very healthy diet. And these are the people in my lab with whom we work every day. Thank you.